We have mapped the 2% of the human genome that codes for proteins with extreme precision, but the remaining 98%, the non-coding regions, has remained largely undecipherable. This expanse of genetic dark matter is the primary orchestration layer of life. It holds the regulatory code determining when, where, and to what degree genes are expressed, yet it actively resists computational modeling. The barrier has long been a strict mathematical trade-off. To model genomic sequences, algorithms had to choose between a wide context window or high-resolution outputs. Because of this, predicting the molecular consequences of genetic variations outside the easily mapped exome has been highly constrained. Standard diagnostic tools frequently hit a wall here, failing to identify pathogenic variants hidden deep in these unmapped regions. Google DeepMind developed alphagenomes specifically to solve this exact context resolution bottleneck. This bar chart shows alphagenome achieving an area under the receiver operating characteristic of 0.91 in variant effect prediction. It outperforms the previous state-of-the-art score of 0.84, while reaching a 0.87 correlation for cage tasks. This jump in accuracy is not the result of brute force parameter scaling. It comes from a novel hybrid neural network architecture. By restructuring how the model reads DNA, alphagenome allows researchers to conduct in silico mutagenesis across the entire genome at a massive scale. To understand how it achieves this clinical utility, we have to look at how the architecture processes 1 million base pairs simultaneously without losing single-nucleotide precision. The previous baseline model, Informer, relied on a standard transformer-based foundation to map genomic interactions. It was constrained to a context window of roughly 200 kilobases and crucially produced outputs in 128 base pair bins. A 128 base pair bin is mathematically too coarse. It obscures fine-grained features like individual transcription factor binding motifs and exact splice junctions. AlphaGenome corrects this, starting with its first architectural stage, a UNet encoder. This encoder directly ingests a massive sequence of over 1 million base pairs. It applies multi-resolution feature extraction, scanning the sequence for both immediate 10 base pair patterns and much larger structural signatures. The raw sequence then compresses down through the network's layers into a dense, manageable mathematical dimension. Crucially, this specific UNet compression does not destroy or blur the local motif data. The sequence is now fully compressed, retaining its local high-resolution markers and prepared for handoff to the next layer. The UNet solves the input scale problem by digesting the massive context, but the system now relies on a different mechanism to identify relationships across that vast linear distance. The compressed data moves from the UNet encoder into the transformer backbone. Here, the model calculates the three-dimensional genomic architecture of the cell. It utilizes cross-attention to route distal genomic sequences and calculate the influence of widely separated data points. This allows the transformer to map precise regulatory interactions over extreme distances, predicting how a distal enhancer bends to contact a promoter 100 kilobases away. Legacy models failed to detect these connections because a 100 kilobase loop structure often exceeded their maximum context window. AlphaGenome's 1 million base pair window easily swallows the entire enhancer-promoter loop, providing the full regulatory context. The transformer maps all of these global relationships and outputs them as a dense latent representation. While the complex 3D interactions are mapped, the data remains locked in a compressed latent space and must be reconstructed to be clinically readable. This reconstruction requires the final architectural stage, the convolutional neural network, or CNN decoder. The CNN decoder takes the transformer's latent representation and upsamples it, expanding the compressed data back into detailed output tracks. As the data exits the decoder, it achieves a critical return to single-nucleotide resolution. The model simultaneously generates predictions across 11 distinct molecular layers. This includes specific tracks for cage transcription start sites, DNase hypersensitivity, and ATAC-seq chromatin accessibility. Analyzing multiple output modalities at once provides a highly robust cross-verified view of genomic activity. This hybrid architecture successfully breaks the historical trade-off, outputting high-resolution functional predictions across a massive 1 million base pair context window. The practical, real-world application of this architecture is in silico mutagenesis, or ISM. We can test this by analyzing a known anomaly in the TAL1 gene. Specifically, this is an oncogenic insertion impacting CD34-positive common myeloid progenitors. The computational methodology begins by running inference on the healthy reference sequence to establish a baseline. Next, the system runs inference on the mutated alternate sequence, overlaying the results to isolate the exact visual differences. By calculating the delta across the predicted tracks, the model mathematically identifies the pathogenesis caused by the mutation. This precise detection relies entirely on the single base pair resolution built in the CNN decoder. A 128 base pair bin would have missed the insertion entirely. AlphaGenome empirically predicts the multimodal functional impact of a novel mutation before a single pipette is lifted in a laboratory. Beyond the theoretical model, the biotech sector is rapidly integrating AlphaGenome into production diagnostic pipelines. This open-source response includes a direct port to PyTorch, developed by the Kundati lab. This port achieved a critical engineering milestone, numerical equivalence to the original JAX model, validated with a Pearson correlation of 0.9999. This PyTorch parity allows seamless integration into the existing software architecture of thousands of biotech startups. Engineers then built a Model Context Protocol, or MCP, server node, allowing large language models to interact with the genomic model via natural language. Wrapper platforms like VariantIQ and 3Billion's Gebra utilize these components to handle complex pre-processing and multimodal column mapping for the end user. This unified stack effectively turns a raw VCF sequencing file into a high-throughput clinical variant interpretation tool. With AlphaGenome at the base of this stack, the work of computational biology has moved past simply cataloging sequences to engineering functional, high-resolution predictions for every base pair in the genome.